At the most fundamental level of human existence, there is a biological process unfolding inside the female body that determines whether life itself can continue beyond a single generation. It is silent, invisible, and yet governed by one of the most precisely regulated endocrine systems known to biology. A system where hormones, neurons, and cells coordinate with extraordinary precision to produce a single, viable gamete prepared for one of the most critical events in nature. Fertilization. This is not simply reproduction, it is molecular orchestration, endocrine timing, genetic division, and cyclical biological architecture working as one integrated system. And it all begins in the brain. Deep within the hypothalamus, specialized neurons release gonadotropin-releasing hormone in a rhythmic, pulsatile pattern. This signal is not constant. Its frequency and amplitude determine the entire reproductive state of the female body. These pulses travel to the pituitary gland, a small endocrine structure located at the base of the brain. In response, the anterior pituitary secretes two key hormones into the bloodstream: FSH, follicle-stimulating hormone, and LH, luteinizing hormone. Together, they regulate the ovarian cycle through a tightly controlled feedback loop, the hypothalamic-pituitary-gonadal axis, a system that integrates neural signals, endocrine responses, and ovarian function into a unified biological rhythm. FSH initiates the development of ovarian follicles. Each follicle contains an immature oocyte surrounded by supporting granulosa cells. At the beginning of each cycle, multiple follicles begin to grow, but only one typically becomes dominant. This selection process is not random. It is determined by subtle hormonal gradients, receptor sensitivity, and cellular competition. Inside the dominant follicle, the oocyte begins one of the most important processes in biology: meiosis. Genui. Biology_learning_block. Type _ID, meiosis. Unlike mitosis, meiosis reduces the chromosome number by half, producing a haploid cell capable of fusion with sperm. But in females, meiosis is uniquely regulated. It begins before birth, then pauses for years, sometimes decades, arrested at a specific stage until hormonal signals resume the process. This long pause is one of the most extraordinary features of human biology, because it means that the potential for life can remain suspended in time, waiting for the correct hormonal and physiological conditions to resume development. As the follicle grows, it becomes increasingly complex. Theca cells surrounding the follicle respond to LH and begin producing androgens. These androgens are then converted by granulosa cells into estrogens through enzymatic activity. This local hormone production creates a self-amplifying environment that supports follicular maturation. Estrogen levels rise steadily throughout the follicular phase, and this rise has systemic effects. The endometrium begins to thicken, preparing the uterus for potential implantation. Meanwhile, estrogen exerts feedback control on the hypothalamus and pituitary, fine-tuning FSH and LH secretion. But the system is not linear. It is dynamic, responsive, adaptive. As estrogen reaches a critical threshold, feedback switches from negative to positive. This triggers one of the most dramatic events in the female endocrine cycle: the LH surge, a sudden spike in luteinizing hormone that transforms ovarian physiology within hours. This surge induces the final maturation of the oocyte, resumption of meiosis, and ultimately, ovulation, the release of a single mature egg from the follicle into the fallopian tube. At this moment, a structure that once supported growth becomes something entirely different. The ruptured follicle transforms into the corpus luteum, a temporary endocrine gland that secretes progesterone. Progesterone is essential. It stabilizes the endometrium, maintains uterine receptivity, and suppresses further follicular development during the luteal phase. This ensures that the system operates in cycles rather than continuous overlapping events. If fertilization does not occur, the corpus luteum degenerates, hormone levels fall, the endometrium sheds, and the cycle begins again. But if fertilization occurs, the system shifts entirely. The developing embryo produces human chorionic gonadotropin, a hormone that signals the corpus luteum to continue progesterone production. Preserving the uterine environment, maintaining pregnancy, and preventing the restart of the ovarian cycle. This early communication between embryo and maternal system is one of the most delicate molecular dialogues in biology. But beneath this visible cycle lies an even deeper layer of complexity: the regulation of gene expression within ovarian cells. Hormone receptors embedded in cell membranes activate intracellular signaling cascades. Second messengers such as CAMP alter transcriptional activity. Enzymes are upregulated or suppressed. Steroidogenesis is precisely controlled at multiple enzymatic checkpoints. Every stage depends on molecular feedback loops operating at different timescales: seconds, hours, days, weeks. The female reproductive system is not simply cyclical, it is multi-layered temporal biology, a system operating simultaneously across hormonal, cellular, and genetic dimensions. Energy metabolism also plays a critical role. Developing oocytes require enormous metabolic investment. Mitochondria within the oocyte are maternally inherited, making their quality essential for early embryonic development. Granulosa cells regulate nutrient exchange, ensuring that the oocyte receives the substrates necessary for maturation without exposing it to oxidative stress. Because unlike many other cells in the body, the oocyte must preserve its integrity for future life. Not immediate function, but potential function. Meanwhile, the surrounding endocrine environment continuously adjusts based on internal and external cues. Stress, nutrition, sleep, and overall metabolic state can all influence the hypothalamic-pituitary-gonadal axis, suppressing or modifying ovulation timing. This ensures that reproduction only occurs under favorable physiological conditions. From an evolutionary perspective, this is not incidental. It is essential, because successful reproduction requires environmental stability, and biological systems have evolved to integrate environmental information into hormonal regulation. At the molecular level, every step of this system is governed by receptor-ligand interactions, intracellular signaling pathways, and gene regulatory networks. Cells do not act independently. They respond to chemical signals with precise biochemical programs encoded in their DNA. And yet, despite this complexity, the system remains remarkably robust, capable of functioning across decades of reproductive life, adjusting continuously, adapting to change, maintaining balance, until eventually, ovarian reserve declines, follicular response decreases, hormonal cycles cease, a process known as reproductive aging. But even this is regulated, gradual, and biologically orchestrated, not abrupt failure, but a controlled transition. Throughout all of this, one principle remains constant. The system is not designed for individual events. It is designed for continuity, for timing, for precision, for the possibility of life emerging from a single cellular fusion event. And perhaps the most extraordinary realization is this: inside the female body exists a biological system capable of coordinating brain activity, endocrine signaling, cellular differentiation, genetic recombination, and tissue remodeling, all for the creation of a single moment of potential life. Not through intention, not through awareness, but through molecular regulation operating with breathtaking precision. A system that has sustained the continuity of human existence for hundreds of thousands of years and continues to do so silently every single day.